Welcome to this session on macromolecules and introduction to biomolecules present in the living system. We see around us in our surrounding, which is made up of animate and inanimate matter. Animate or living organisms are those which respire, respond, reproduce, whereas inanimate systems do not do all of these. If we look into what they are made up of, both may have similar type of biomolecules. But this is a very important factor. They behave very differently, each system. Now let us focus. The molecules present in the living system are called as biomolecules. And to study them with respect to their emphasizing on their structure, properties, biological or physiological function, and industrial or commercial significance is biochemistry or macromolecules. Now, as I said that the living system are made up of biomolecules. Now let us understand what these biomolecules are. Biomolecules are those molecules or chemical substances present in the living systems, and mainly they comprise of carbohydrate, protein, lipid, nucleic acids. Now, how can we ensure that these are present? This can be done by very simple identification test. Now, Mulisch test is a simple test which can be done for the identification of carbohydrates. Here we take the sample. To it we add the Mulisch reagent, which comprises of 10% alpha naphthol and concentrated sulfuric acid. Carbohydrates react with Mulisch reagent to form a reddish-violet zone indicating their presence. We can identify the proteins by performing the biurate test. We can take the sample. To it we can add the biurate reagent, which is mainly alkaline copper sulfate. The proteins, if they are present, react with the copper sulfate to form a purple colored protein copper complex. If purple color is there, it indicates the presence of proteins. Similarly, we can elucidate the presence of lipids by performing a simple translucency test. On a paper, you put the sample, and if the paper becomes translucent, then we can confirm the presence of a lipid. Now, here I have a short video which shows we have taken the samples originating from various sources, the plant, animals, and we have done I have shown to you a simple experiment of Mulisch and biurate test and the paper test, which indicates the presence of all these carbohydrates, proteins, lipids. Let us see the small video. So now everyone has seen the experiment demonstration of the presence of carbohydrates, proteins, and lipids from the various sources of animal plant extracts. Now let us try to understand these biomolecules. Let us start with the very first one, the carbohydrates. What are carbohydrates and how are they defined? Carbohydrates are defined as polyhydroxyl aldehydes or ketones, or those substances which on hydrolysis give them. They are also commonly called as saccharides or sugars. Constitutionally, they are basically made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now, after knowing the definition, let us concentrate on towards the structure of carbohydrates. As we defined that carbohydrates are polyhydroxyl aldehydes or ketones, so they essentially comprise of a hydroxyl group, not just one, bare minimum 2. It can be more also. So therefore, then only it can be called as a polyhydroxy compound. So in every carbohydrate, there essentially needs to have at least 2 hydroxyl groups or many hydroxyl groups. So many hydroxyl groups, and along with it, a carbonyl group is attached, either in the form of a ketone or in the form of an aldehyde. Now let us see this with an example. Now here I am representing a aldehyde containing carbohydrate. So essentially, it contains an aldehyde group, CHO, and many hydroxyl groups, OH, OH, OH. So such a carbohydrate containing an aldehyde group as well as polyhydroxyl group, it is called as an aldehydic sugar or an aldose. Now let us go to another type of category of carbohydrates wherein it compromises in its structure many OH groups as well as a carbonyl group. So when the carbonyl group is present in the form of a ketone, as you can see it out here, a ketone group along with many OH groups, it is called as a ketonic sugar, or it's also referred to as ketose. So this carbohydrate is the basic structure to be called as a carbohydrate. Now after knowing the basic structure for carbohydrates, let us proceed further to what are the functions of carbohydrates, why are they so important to us for us to study. Now let us look into their functions. Glucose or glycogen are the major source of energy. For any activity of any living system, if it has to perform any activity, it needs energy, and this is derived from the carbohydrates. The next category of carbohydrate, let us take example cellulose. Now cellulose, you all very well know, it is present in the plant systems. In the plant cell wall, cellulose, the main function of it is to impart strength and rigidity to the plant cell so that it can sustain the osmotic pressure developed inside the cell. Another category of carbohydrate, that is mucopolysaccharide, act as lining or protect the interior of many organs, for example the stomach, where the pH is around 1 to 2, highly acidic. Otherwise, without the presence of this mucopolysaccharide, probably the stomach would have eroded. Another example is hyaluronic acid. Hyaluronic acid is a polysaccharide which is present in the synovial fluid, which is present as a lubricating agent in between the joints, and thus it lessens the resistance developed due to friction and avoids the inflammation of the joints. Next example is heparin. Heparin is a very good natural blood anticoagulant and maintains the free flow, laminar flow of the blood flow in the blood vessels. Another example, glycoproteins. They act as antibodies providing immunity in our system. Similarly, there are many such carbohydrates having industrial significance in various sectors such as pharmaceutical, medical, engineering sectors.